castle where the digital dreams come alive. CESCG takes place from May 1 to 5, from visualization to the virtual space. Scientific sessions take their place.
Hello, I'm Patrick from Slovak University of Technology. I am bachelor student under supervision of Vanda Benešova, and my topic is deep learning-based segmentation and classification of histological colon cells. At first, our motivation is to create automated support for pathologists to diagnose inflammation process in Crohn disease and ulcerative colitis. Right now, we are cooperating with doctors from Bory Hospital. Our goal is to diagnose and create a rule-based processing architecture based on a deep neural network. We really want to implement feedback from doctor, and we want to enhance and make more efficient annotation process with this architecture. This research started when doctors from Bory addressed us. We had big problem because we didn't understand what they want. We didn't have same language. Because of this, we implemented human in the loop approach into the system, where my colleague Robert came to this problem, to resolve this problem. And it can be seen that at the first, we found out that we have a lack of data. We had too little data, and we was not able to do exactly what they wanted. We told them that first process is to gather data and create more annotations. Overview of our approach started when we trained our model with public dataset. This public dataset is conic dataset. We used it for transfer learning. After that, we use this model for creating of pseudo-labels. These pseudo-labels are then manual revision of these pseudo-labels by pathologists were performed. Two types of this revision can be done: correction of selected pseudo-labels or optional rule explanation of this correction. With this data, new corrected and origin data, the model is trained. After this, our model is evaluated by annotated data from doctors and pathologists. Now I'll tell you something about our proposed solution. How I said, we have a lack of data. Because of this, we implemented transfer learning with a conic dataset. That's first dataset which we used. Our second dataset is made of 4 whole slide images, which we got from pathologists. Our system is made of 3 parts. First part is segmentation module, the second part is filtration module, and third part is classification module. The main part of our filtration module is adjustable filters. These filters are extracted from feedback from doctors, about which I will say later something more. First module, segmentation module, we experimented with 3 architectures: unit architecture, AttentionUNET architecture, and residual unit architecture. Results of DICE score of this architecture was quite similar, but in the pictures, which can be seen here, mostly on the bottom side of the image, which are predicted masks, can be seen that AttentionUNET performed the best. Because of this, we used for our system this AttentionUNET. Second module which we used was a filtration module. This filtration module implement in our system interactive tool. This filter is adjustable by thresholds, how I said, and it use specific filters. Right now, in our system are implemented 3 filters: area-based filter, color-based filter, and circularity-based filters. These filters are implemented out of feedback from doctors. They said they are really interested while diagnostic of Crohn disease and ulcerative colitis in cells which are bigger, which are darker, and which are in a specific shapes. Here can be a seen example of area-based filter. Red parts are filtered cells. This filter can show that it worked and it filtered small cells. This can be also set by threshold. We can filter bigger cells if we want. Second, filter is color-based filter. We can see that non-filtered images are mostly darker images. And last filter is shape-based filter. On the right side, on the mask, segmentation mask, can be seen that more longitudinal cells are filtered. With a combination of these filters, we can extract only cells which are really important for pathologists, and we don't need to show to the pathologist cells which are not that important from them. Classification module, we also tried different architectures for this. Few architectures were ordinary convolutional neural network. Second was VGG16, and the last one was ResNet 34. The accuracies of these architectures was quite similar. That's because we trained our models only on conic dataset. This dataset is not in a good resolution, so it's not able to extract all of the features needed for classification. Here can be seen output of our classification module. So it can be seen that plasma cells and eosinophils are predicted quite well. Problematic are lymphocytes, because lymphocytes and plasma are really similar cells. They are, both of them are lighter cells, and this is done because we didn't have that good resolution of the training data, but this problem will be solved after annotating more data by pathologists and training model with these data. After that, we also visualized attention of our classifier. On the left side can be seen that sometimes our model is looking at correctly at the cells in the middle, and sometimes it's not looking quite on cells which we want. Because of this, we implemented attention module into our classifier, which is combination of image patch with segmentation mask. This will show architecture where exactly on which pixels is the cell. Improving of our model performance, we found out when we converted both images from a custom dataset and conic dataset, when we showed it in LAB color scheme, we can see that L channel histogram is different in both of them. L channel is lightness. That means both of the datasets are not in the same color range. Because of this, we needed to create or perform normalization of this channel. We calculated average L channel over all images and performed this normalization over this data, and we trained a model after with this data. Second improvement is that we found out that only 400 out of 600 annotated cells by pathologists predicted our segmentation model correctly. Because of this, we needed to train our model with this data, but all of cells from the image were not annotated. Doctor annotated only one cell, and when we train our model with this only one cell, it can confuse the model. So we needed to create pseudo-labels and add these pseudo-labels with annotations from pathologists together and train this model with new dataset. This new dataset was not huge, so we needed also perform augmentation of our images to have more data to train with. Presentation before showed a very good annotation tool, Annotate. We also implemented our approach into this tool. So with this tool, we want to perform feature selection. This feature selection is for information retrieval. This information retrieval is then used for derivating of filters, which we then use in our filtration module. It can be seen here that we can select cell, and pathologist or domain expert will note some description about this cell. Then after that, we will process these descriptions, and we can extract important new features of the cells. Second part is active learning. We implemented active learning into this system. We iteratively select the most informative data. Right now, we have only implemented most certain predictions and most uncertain predictions. It can be seen here that when the model, we predict these most uncertain and most certain predictions, and after that, the pathologist needs to correct this, and we can see this process right here, where the pathologist correct this exact cell. We implemented human-in-the-loop process into our research. We create interactive feature-based system, and we fine-tuned our model, and we also implemented this system into the annotation tool Annotate. Our future work is to create active learning more precise and perform and implement better techniques of active learning into our system. We want to enhance model performance and identify new filters. Thank you for your attention, and now it's time for questions.
Thank you, Patrick. So maybe there is room for a question from the audience.
Thank you for the presentation. So on one of the slides you mentioned that there is this feedback loop from the experts that they provide some sort of rules that you then incorporate into this filtration. So that sounds very difficult, right? So how do you do this? I assume that the rules that they provide are in some vague natural language. So what is the process of incorporating those rules into the system that we probably saw the result, but how do you validate it, and how do you communicate with the doctors to do this?
Thank you for the question. Because of this, we have this human-in-the-loop approach, and we have our colleague Robert. He talked with pathologists, and he found exact problem and what we are solving. For example, right now we are solving Crohn's disease and ulcerative colitis, and doctor said, for example, longitudinal cells are mostly in a muscle tissue, and that's exactly cells which we don't want to predict or classify because it's not important for this disease. Then they wanted more darker, for example, neutrophils are very dark cells, and these cells are extremely important for them right now with this disease. Because of this, we find out that we need to create a color-based and shape-based. Area-based first filter, which filtered only small cells, were there because sometimes the segmentation module create false positive predictions, and we wanted to filter some of them. This is process, for example, how we implemented these 3 filters. After that, we'll have more sessions with doctors, and we will try from a description how I showed in this slide that he will sit and write some description about this cell. That I choose this cell because it's long, it's light, for example. And from these notes, we will gather this information.
Thank you.
Thank you.
Okay, thank you very much for your presentation. I would like to ask about slide 13 with where you were comparing the saliency map. Okay, not 14. You were comparing the activations at 12. So I would like to ask, how was the saliency map created? It's from ground truth data, or like what is the saliency map in this case?
Okay, I extracted weights from the model because all of the modules I created custom. So I didn't implement functions. So in last layers, I exported these weights and performed with these weights prediction, and it created saliency map.
Okay, thanks.
Thank you.
Okay, thank you very much, Patrick.
Thank you.
Swing to the beach where the pixels play. From rendering to VR, it's a pixel